Last time, I don't remember exactly how much of this slide I had talked about, but this is where I had left off, I'm sure. So I had started by talking about lipoproteins, and this is essentially the last concept we need to cover before I draw you the big picture, which will start in the next slide. Lipoproteins are, as I think I had introduced last time, they're these large clusters, as it says, of cholesterol, triglycerides, monoglycerides, fatty acids, etc., that have this coating around them of apolipoproteins, hydrophilic proteins that have hydrophobic underbellies, and then other phospholipids, glycerophospholipids, that help them stay in solution in your blood. So there are 5 categories of them, and they are separated by their density. And when I say separated by their density, when you take a whole blood sample and you spin them down, they are going to segregate. So when I say spin them down, I put them in a centrifuge, and you separate the blood into plasma and whole blood and the white blood cells and the buffy coat and all the different components separate out. The lipoproteins will separate based upon their densities. So the least dense of them are called chylomicrons. Also ULDL, ultra low density lipoproteins. And these ULDL molecules are the vehicle, as we'll see in a moment. They are the vehicle for getting lipids out of the gastrointestinal system and into the rest of your body, into the bloodstream. And then the rest of them are essentially distributive within the bloodstream. There's very low density, intermediate, low, and high density lipoproteins. We're going to see what each of them does one at a time. So the largest of them, and their density, of course, tracks along with their size. So the ULDLs are the largest of them, and the biggest ULDLs or chylomicrons are up to 1,000 microns. How big is a 1,000 microns? Anyone remember their metric conversions? So a micron is 1,000th of a millimeter. Micro versus milli is an order of 3. So 1,000 microns is 1 millimeter. If you think of a centimeter as, at least on my finger, being the width of one of my intermediate fingernails, and a millimeter as maybe being the thickness of my fingernail, maybe a little bit less than a millimeter, that's the size of one of these chylomicrons. So sort of the thickness of a fingernail. That means that chylomicrons are, in fact, visible to the naked eye. They're one of the smallest possible structures that your naked eye can clearly distinguish. All right, so these are the lipoproteins. Let's see how they work. We just had breakfast. Who had a healthy, delicious breakfast today? Good, me too. A few of you guys don't eat breakfast, huh? After the class. Okay. All right. Well, after class, you're going to have breakfast. I had breakfast before, and I had some delicious whole-fat yogurt with a little bit of fat globule in there over my oats. And that fat globule is in my gut right now. And it is not emulsified. It is not anything. It's just in the lumen of my intestine. And when I say lumen, that's just the inside of the giant gut tube that is a human being. All right, just inside the contents of the intestines. Now, the cells that line the inside of the intestine are called enterocytes because the word enteric refers to the gastrointestinal system, your GI tract, specifically the intestines. So we have these epithelial cells that line the intestinal lumen, the enterocytes. All right. This fat globule gets converted into an emulsion droplet by the bile salts. And here is glycerylcolchicine that I had talked about the bile salts last time. I showed how there was a hydrophobic and a hydrophilic side. You can see here how indeed there is a hydrophilic side, all this red and blue being nitrogen, all on the same side, and then this really greasy side here. It still has the cholesterol backbone, but if you look at it closely, particularly those who looked at cholesterol last time, you'll notice that the overall 3D conformation of that ring structure is quite different because it has had two of these methyl groups stuck on here instead of hydrogen, which force it into this sort of like curled up formation that sends all of these hydroxyl groups to the same side of the molecule. So the bile salt and the glycerophospholipids and monoglycerides, etc., form these emulsion droplets in the intestinal lumen. Pardon me. Thanks. Yeah, something's getting me. The lipase, pancreatic lipase, which we've talked about, breaks the triglycerides up into monoglycerides and fatty acids, which form smaller structures called micelles. And micelle is a generalized term that means any kind of lipid that has been suspended in an aqueous environment because of some sort of emulsifier, surfactant, amphipathic molecule. Those are all the same words. Detergent. So we essentially get these smaller structures called micelles after the larger triglycerides are broken down into their constituent fatty acids. These micelles spin off the fatty acids and monoglycerides, which are absorbed by the enterocytes. That's what actually goes into the enterocytes. Free fatty acids are absorbed by the enterocytes. These free fatty acids are then packaged into ULDL or chylomicrons by the enterocytes. So this involves adding cholesterol to them. This involves adding various apoproteins to them to help emulsify them. Once again, create these lipoproteins, these large 1 millimeter up to 1 millimeter diameter balls of lipid and protein that are able to once again go into solution in an aqueous environment. Yeah, Damon.
Like when you have your emulsion droplet, what?
The action of lipase. Yeah, lipase is associated with the emulsion droplets, hydrolyzes the triglycerides into free fatty acids. And when you have these smaller molecules, the free fatty acids, you're able to get a smaller droplet called a micelle. You can't make a micelle in terms of the nomenclature that we're using here with the triglycerides. Any other questions? Okay, so in your intestines, you may know this, and we'll see this in probably on Wednesday. The lining of your intestine has all of these big ridges up and down that increases the surface area of the intestine. All right, within the lumen of your intestine, there's all these ridges, and that you want high surface area because the surface area is how you absorb the nutrients. The higher the surface area, the more surface area for absorbing nutrients. And within the body of those ridges are these structures called lacteals. Lacteals. Now, a lacteal is part of your lymphatic system. And the lymphatic system is basically part of your immune system. It's the part of your immune system that shuttles white blood cells around that aren't in your blood. It runs parallel to your circulatory system, but it is not your circulatory system. And the lymphatic system doesn't have a pump the way your circulatory system has. Instead, it has these lymph nodes, and anyone who's ever had a cold has probably felt their lymph nodes in their neck, or if you've ever had an infection somewhere, you've felt a lymph node in your elbows, behind your knees, your groin, maybe behind your neck, sometimes by your ears. They'll get big. And it's those lymph nodes that when you move your body kind of compresses those lymph nodes and sort of sloshes the lymphatic fluid along that lymphatic system. All right, so just kind of by squeezing on those lymph nodes. This is why bedbound people don't, because they're not moving, they're not actually circulating lymph as well as they could. So bedbound people actually can have a harder time clearing an infection in an area. So the ULDL, the chylomicrons, first pass into the lymphatic system through these lacteals. They go through the lacteals and then pass into this complex network, this duct system of lymphatic ducts that go throughout your body. And they converge on something called the thoracic duct. It's this duct that, I guess I could have put a picture up here to show you what it looks like, but there's this duct that is in the thorax. It comes out here on the left-hand side underneath your collarbone and attaches eventually to the circulatory system. So ULDL passes, leaves the enterocytes, and they drain into the lymphatic system. These ULDL then travel through the lymphatic system all the way to the thoracic duct and pass through the thoracic duct into the bloodstream. Once they're in the bloodstream, these ULDL molecules are free to deliver the fatty acids and the triglycerides that they're carrying primarily to the liver. They're trying to get to the liver. All right. In the liver, we begin to produce the other lipoproteins. And the first one I'm going to talk about, and in some ways the most important, is HDL, high-density lipoprotein. It's produced in the liver, and then it gets released by the liver into the circulatory system. HDL is often called, it's sort of an overly simplistic appellation, but they call it the good cholesterol, right? So if you've ever heard about LDL or HDL before, it's probably in the context of good and bad cholesterol. And HDL is what we call good, is what they refer to as the good cholesterol. So what does it do? Well, it does a number of things. One of the things that it does is it interacts with chylomicrons in the bloodstream. And since it's high-density lipoprotein, it means that there are more proteins and less lipids in it, all right? And those proteins can get transferred from HDL to chylomicrons en route to the liver.
Aren't lipids more dense than?
No, because if you think about that, so water sinks, fat floats, right? And that's because water or any kind of hydrophilic polar molecule has that polar interaction that pulls them together and increases the density of them. So protein tends to be denser than fat, for sure. So the HDL, it delivers these proteins to the chylomicrons, the ULDLs, and in so doing, one of the things that happens is that the chylomicrons begin to spin off triglycerides and fatty acids. And as it spins off these triglycerides and fatty acids into the blood, they are getting taken up by the peripheral tissues. And then the next thing the HDL does is it's able to either pick up cholesterol or deliver cholesterol to tissues or lipoproteins, as we'll see in a moment, other lipoproteins, depending upon their need. Does the tissue need more cholesterol? Well, it delivers cholesterol. Does it have excess cholesterol? It will pick some of that up. All right, and while it does, as it does all this, it eventually returns to the liver and is recycled by the liver. All right, so that's HDL. Yeah, Damon.
So you said like after. When manages.
HDL is going to, as it's going through, it's interacting with the different lipoproteins.
Yeah.
So like the chylomicron, and it will deliver proteins to the chylomicron, which is going to affect the structure of the chylomicron. The chylomicron is going to spin off fatty acids and triglycerides. Yeah, and the tissue, so the tissues can then absorb them. The HDL will also directly pick up cholesterol from tissues if a tissue has a surplus, or it will deliver cholesterol to a tissue if it's deficient and it needs some. And this is kind of a biophysical property of this lipid transfer. All right. Next, we have VLDLs that emerge from the liver. All right. And VLDLs, very low-density lipoproteins, are also delivering cholesterol and triglycerides to tissues. So now you can see there are two mechanisms for getting fatty acids and triglycerides. There's exogenous and endogenous, meaning there's stuff that came from the digestive system that's being delivered by the ULDLs, and there's stuff endogenous or from inside you that's coming from the liver via the VLDLs. All right, so there's sort of two parallel pathways for getting fatty acids and triglycerides to your body, your peripheral tissues. One from the gut via the ULDLs, and one from the liver via the other lipoproteins. And HDL is moderating all of this. So in much the same way as HDL interacts with chylomicrons, it interacts with VLDL as well. You can see by the little white arrows that I put there. It's interacting with VLDL and causing them to deposit the triglycerides and the fatty acids. As VLDL loses lipids, they become an IDL, intermediate density lipoprotein, which continues to spin off triglycerides and fatty acids. Again, interacting with HDL in that process. As they lose lipids, they eventually become LDL. And the job of LDL is to bring excess triglycerides and fatty acids to the liver. All right, so the VLDL and the IDL, they're bringing endogenous triglycerides and fatty acids to the tissue, and LDLs are bringing the remnants back to the liver. So when you look at this, you can see that there are essentially two ways of delivering the triglycerides and fatty acids, one from the gut via the chylomicrons, one from the liver via the VLDL and the IDL, and both of them are regulated by HDL. So the liver's ability to regulate the lipid content of the blood and delivery to the tissues and cholesterol content of cells is based upon its ability to regulate HDL. How much HDL is your body making, right? And so this is why HDL is, the more HDL you have, that it's seen as being a good thing. Now, why would LDL be a bad thing? So it's bringing triglycerides and fatty acids to the liver. Well, if there is too much excess triglycerides and excess fatty acids in the blood, there's going to be more LDL than your body can really handle and absorb by the liver. And so what happens is these cells called macrophages, macro being large, phage meaning eat. It's these large, it's like the amoeba that ate Philadelphia kind of thing. There's these giant they're part of the immune system, these large immune system cells that sort of blob their way along through your body. They take exception to LDL, and they will consume LDL. If there's too much LDL in your body, macrophages consume LDL. Once these macrophages eat the LDL, they become glutted with the lipids that were in the LDL to begin with. And they transform from macrophages into what are called foam cells. That may sound kind of delightful, but it's really not a good thing. Because if there are too many foam cells initiate an inflammatory response, which causes the foam cells to deposit on the walls of your arteries in your body. So this is what leads to atherosclerosis. If there's too much LDL and not enough HDL, so not enough of the triglycerides are getting dumped to the peripheral tissues, you can lead to an overabundance of foam cells and a hardening of your artery. All right. Also, I didn't put on this diagram, but does anybody have like a familial history of, like, a lot of people in your family have had heart disease? I don't know. My dad's whole side of the family is that way, and I've never had genetic testing on. They've never had it, or they're all dead now. But I've never had it to know whether it's the case or not. But the arrow here that shows LDL going into the liver right at the end, people with a familial disposition to hyperlipidemia or heart disease, coronary artery disease, those people often have a genetic problem with that part of the pathway. Like the liver is not able to take up LDL as readily as it should. So that can be a problem that many people have, and it's persisted in the gene pool because people used to die from cholera or whatever by the time you were 45, well before hyperlipidemia would give you heart disease. But we're living longer now, and our diet has changed as well, and so this has become a major problem that I guess evolution hasn't really taken care of yet. So that's how fat passes through your body. That's the story. Does anyone have any questions about that? No? Yeah, what?
Sorry, when that goes, like when you put that on Moodle, do you know if that whole process will be shown?
Yeah. Is this on the version that's on Moodle? Is this in there? I think it is, or maybe it's not. Yeah, when it goes to Moodle, yeah, every single one of these steps is in there. I don't use animations. I do individual slides. That's a lesson for you. Don't use PowerPoint animations. They'll fail on you. Okay, so LDL to HDL ratio. Let's talk about it. So, in general, people who, if you look at that ratio of LDL to HDL, if it's low, so around between 2 to 4, then you're at low risk. All right. If you are between 4 and 7 in your blood titer, that's that makes you an average American. That's typical. All right. And above 11 puts you at higher risk of coronary heart disease. Let's look at a little bit at the specific composition of HDL versus LDL. If we look at what's on the surface, we see significantly more proteins in HDL. That shouldn't be surprising. It's high density. Its job is to deliver proteins to some of the other lipoproteins. And it has less cholesterol in it. It is one of its main jobs is as a cholesterol scavenger from the tissues. So there's more room in it for that. What else do I want to say here? Yeah, so high rates of HDL correlate with low rates of atherosclerosis, and the opposite is true of LDL. Yep, I guess that's all that I actually have on that slide. Then this is, I think, my last slide here. So if you look at above this 11, so here on the bottom are numbers from, I said 7 was the outside of average or normal risk, and then above that became higher risk, and this goes into the really high risk. So if you look from like 8 to 32 LDL ratio, 7% of those people show a risk of coronary artery disease, all the way up to, there are people out there with 80 to 200 ratios of LDL to HDL. That would be probably a person who's either eating sticks of butter, or you have a 96% chance of coronary artery disease, and that could also be a person with a genetic predisposition, the way I spoke about it earlier. So, I have this last slide about adipose tissue. Class is over. So next time I will talk about my daughter here and baby fat, and then I'll read through the summary slides on fat, and then we'll start in on the GI tract. Okay?